preservation. We are go for using a biotechnology to reduce the emissions such as GHI, greenhouse gases emissions. We can reduce the waste of the by using bioremediation techniques. Can conserve the land also. We can conserve the soil also by using biotechnological tools. One of the emerging trends are the, especially in the field of the agricultural biotechnology, are the CRISPR-Cas19 technology. Is the recent trends in the or recent technology which are used in the biotechnology. Biotechnology. CRISPR biotechnological tools is one of the direct manipulation in the crop or direct manipulation in the cell. We can using this technique CRISPR Cas19 protein, which are the RNA mediated gene transfer technology, and which is specially we use in the agricultural sector in nowadays. And uh, before we are using a different types of the CRISPR technology, yeah, using a one short segment of the RNA molecule, which are the manipulate at the genome editing in the crop. Previously, we are first going for the genetical modification in the crop by using selective breeding, which are the from the Mendelism, and then we are go for the random mutagenesis.
एवढं एकच आहे तुमचं किती वेळ लागला सरला इकडे हॅलो
Pawar sir, you are not audible.
Invented the transgenic technology to modify the crops by using different types of genetical tools, which are transgenically. One of the product is a BT cotton is one of the product of the transgenic crop, and then now we are moving towards the CRISPR genome editing. CRISPR Cas9 uh, genome editing technique is a recent trends in the agricultural sectors or the crop improvement technology or biotechnology. Uh, then we are synthetic biology also used for the crop improvement in the also the by some bioinformatical tools which are the editing genome editing also used, and to improve as well as the to develop a drought resistant pest resistant varieties as well as the nutrition nutrient enriched types of the food crops which are the used for the uh, in emerging trends in the biotechnological applications. CRISPR Cas19 is technology which are the synthetic biology. Synthetic biology is the future of the agricultural sectors. Is a by using crop demonstration and breeding methods, which are also developed by the in a synthetic biology, as well as the microorganism utilization also in the field of the microbiology by some biofertilizers, biopesticides, which are the and as well as the soil remediations or the bioremediation process also to improve the crop because our soil by using different types of chemical fertilizer, chemical pesticides, which are the totally loses the our soil health. And now we are the some microorganisms which enrich the soil, such as which called as a soil remediation. And by using such biostimulants which are the used for the for the remediation of the soil. And we are also use some synthetic biological tools in the agricultural sectors for the carbon and nitrogen fixations. We are also the maximum carbon nitrogen fixation ability can be improved in the crop by using different types of genetic modifications, which are the done by the in the synthetic biology. And then we are the future perspectives to the climate climate agriculture sectors are also the using some different types of the techniques which are to improve the also the vertical farming as well as the control environmental agriculture also the using in a future directions. And also the we are the biofarming nothing but the pharmaceutical products which are used in the products produced by using different types of the crops which are genetically modified by involving in the or the by some vaccines are also the edible vaccines are produced by the in the farm in the biopharmaceuticals or biopharma. There are some crops are also act as some vaccine producing agents, vaccine production uh, fruits as well as the it's called edible vaccines. And edible vaccines also the future of the uh, pharma agricultural sectors. In agriculture sectors we are then we also use different types of intelligence. It's called as artificial AI as well as the ML, machine learning also in the biotechnology or the agricultural biotechnology in the futures.
नाही तेवढं नाही म्हणे अजून 2-3
My
मिनिटं
my
ते काय होते कोणत्या कामाची एकदा गोडी लागली का माणसाला ते बरं वाटतं आणि
my main research area is PGPR.
विचारलं बुवा
And PGPR is nothing but the plant growth promoting rhizobacteria, which are the very important in the plant growth promotions. And rhizobacteria is nothing but the microorganisms of the consortium of the microorganisms which enhance the plant growth. And the plant growth PGPR and the rhizobacteria nothing but the PGPR is term is developed by the Cooper and the Strauch, which are the in 1981, which are termed the coin the term PGPR are the plant growth promoting rhizobacteria. Plant growth promoting rhizobacteria which are specially present in the rhizosphere of the root narrow zone of the soil, specifically influenced by some of the roots, and which are the populations by the microorganisms or the bacteria called as the rhizobacteria. And three types of the bacteria in the present in the rhizosphere. First type is the beneficial bacteria. Second time is negative impact is called deleterious types of the nature of the bacteria, and the neutral types of bacteria also present in the rhizosphere. The beneficial types of the free living bacteria, soil bacteria, referred to as a PGPR, reported a PGPR. The PGPR specifically some Bacillus species, some Pseudomonas species which are predominant in the PGPR or rhizosphere, and which specifically act as a PGPR for the specific crops. And the types of PGPRs, which are the extracellular PGPR as well as the intracellular PGPRs, and intracellular and extracellular PGPR, which are the definitely used for the plant growth promotions. Extracellular PGPR is also called as EPGPR, and intracellular PGPR called as PGPR. And EPGPRs, extracellular PGPRs in the rhizosphere, rhizoplane surface, and in between the surface of the root. And intracellular PGPR, which are the inside the root cells. And one of the nitrogen fixing bacteria is the rhizobacteria, which are the very good example of the IPGPR or intracellular PGPR. Different types of mechanize the plant growth promotion by the PGPR, a direct method, direct activity, and indirect activities. The direct activities, which are nutrients, which are the recycling. Phytostimulation is the direct types of plant growth promotion or plant growth activity. These are the direct by the nitrogen sources, potassium sources, phosphate, and the different of minerals which are required for the plants, which are directly benefited from the plants by using microbes. It's called direct activity or direct mechanism to plant growth promotion as a PGPR. Also, the some phytohormones which are produced by the bacteria. For example, the indolacetic acids and different cytokinins, which are the used for the plant growth promotion or phytostimulations. Which are the direct activities, and indirect, such as a systemic resistance, abiotic stress, which are also the indirect type of activity, plant growth promotion, and also the against the some types of the abiotic factors, which are the against the uh, some types of the PGPRs which are used for the uh, different types of the indirect type of the activities. Direct type of activities: biological fertilization, biological nitrogen fixation, phosphate solubilization, phytohormone production, says different types of the siderophore productions. These are direct methods, and indirect, these are the some types of the enzymes which are trigger the against the type of pathogens like phytopathogens, and in the antibiotic production by using indirect methods, indirect activity against the induced systemic response. Also, the which types of the some enzymes which are also the damage the pathogens, phytopathogens, and indirectly promote the growth of the plants. These types of the such as chitinase, glucanase, these are the enzymes which are indirectly promote the plant. And some specific genus which are the used as a plant growth promotion or the PGPR, some like Pseudomonas genus, like Rhizobacteria, Azotobacters, and Rhizospirillums, Azospirillums, and the Arthrobacters, Acinetobacters, and Brandyrhizobiums, and Bacillus, some Enterobacters, some Erwinia also used as some biological fertilizers or the biopesticides in the activity. These are the genus which are specially used for the plant growth promotion in the agricultural biotechnology agricultural sectors. My research area specifically on the Pseudomonas, which are the main number of the types of the plant growth promoting activities, such as the by producing a siderophore, by producing uh, different types of antibiotics such as pyocyanin, pyrrolnitrine. These are the antibiotics which are specifically produced by the Pseudomonas, and these Pseudomonas uh, promote the plant growth by indirect as well as the direct methods. And the Pseudomonas, why the Pseudomonas used in the agricultural sectors? Okay, because of Pseudomonas have the rapid growth in vitro production or mass production. And these have rapid utilization of the seed and root exudates. These are the colonize and multiply in the rhizobacterium. These compete with the aggressively compete with the other types of the microorganisms, specifically the pathogens, plant pathogens, and is also adapt the various stress conditions, environmental conditions in the environment. Hence, the Pseudomonas is one of the best candidate for the as biopesticides for the plant growth promotions. Is by using the bio controlling activities. Pseudomonas produce different types of the PGPR allelochemicals. Allelochemical, nothing but the chemicals which are the inhibit the growth of the other type of bacteria by using different allelochemicals which are produced by the Pseudomonas. Siderophore is also the have the direct as well as the indirect activities. It can also the inhibit the growth of the pathogens as well as the heat provides the maximum ferrous or iron to the plant for the promotion. Yes, the siderophore is indirect as well as the direct plant growth promoting molecule or allelochemicals. Then antimicrobial compounds, so n number of antibiotic microbial compounds such as phenazine, different types of phenazines, phenazine 1 carboxylic acid, pyocyanin, pyrrole nitrine. These are the molecules which are produced by the Pseudomonas species or Pseudomonas genus. The mycolytic enzymes also the enhance or also the important role in the plant growth promotion, such as chitinase, beta 1 3
काय
glucanase,
म्हणलं दिलं मला
also the detoxification enzyme, some other
सबस्क्राईबर
also the
राहिले
use
तरी
for the as a
सांगितलं
mycolytic enzyme. Mycolytic enzymes means the which are the inhibit the growth of the uh, especially the fungal phytopathogens, and fungal phytopathogens can be can be destroyed by using a pseudomonas species of their molecules also.
ते ऑर्गॅनिक फार्मिंग
The
सांगितलं ना
parasitism types of the fascination increases the parasitism activity also the done by the
झाले का पाच
pseudomonas species. Pseudomonas, the bioactive molecules has a number of bioactive molecules, pyrroles, different types of enzymes, different types of the fluoroglycanols, another quinolones. These types of the molecules which are produced by the biotechnological applications in the field of the agricultural sectors. The why we are use the biotechnological tools, because the biotechnological tools are the sustainable tools which are without damaging eco-friendly tools, and hence we are the going for the microbes for the betterment of the agricultural sectors. And microbes are the different types of microbes such as Bacillus thuringiensis K1AC genes. We are already transferred from the Bacillus to the cotton, and these are the tools, biological tools or microbial tools, which are important for the production or to increase the yield of the different types of the crops. And specifically, the biotechnological tools also including recent technologies such as you are already read about CRISPR-9 Cas9 techniques, which are the very popular within some days, and which are very important technique to improve the quality of the yield, and hence we increase these types of the crops which are specifically uh, which are the important or crop which are very important. And nowadays we are also the heard about AI, artificial intelligence. Artificial intelligence also the in future is a play important role in agricultural sector or agricultural biotechnology, because agriculture sectors already in some countries also use AI or and as well as the machine learning, which are used for the agriculture productivity enhancements program. We are already one setup of the agricultural biotechnological uh, setup in the field of the in agriculture or the first lab in the agriculture. We our some Maharashtrian scientist or Maharashtrian, we are work on the AI used in agricultural in Baramati. The we are using artificial intelligence to improve the plant yield and as well as the crop yield. Hence, the biotechnological applications or biotechnological tools very important in the field of the agriculture sectors. Hence, the essential for the sustainable agriculture as well as the to identify the commercial and efficient and should the further research understanding the mechanics in the field of the agriculture. We also uh, the efforts in till required for the PGPR, make a PGPR consortium for the and efficient techniques in the sustainable agricultures. And with this, I am at the concluding session. This is a very important session for the emerging trends in the agriculture sectors. Now, I am very thankful to organizers of the this national webinar on the emerging trends in the agriculture biotechnology, and also I am thanks to the my dear friend Professor Anant Koshi for giving an opportunity as a resource person for this national webinar. And I also very privileged to the share my share the session with my respected Dikshit sir. Okay, with this, I will I would conclude my this session.
सर येतं का नाही म्हणलं का मग
If
बोलायला
have
लागलं
any queries, you can ask.
थँक्यू सर थँक्यू सर thank you very much. Our principal, Honorable Dr. Nimburu sir, is here as a chairman of this session. Out of his very busy schedule, he got ready to be chairperson of this session. He is on the way to join. I request Honorable Principal to conclude the session. Meanwhile, if there are any questions from the delegates, I request the delegates, you can unmute your mic and ask the questions to Honorable Resource Person Pawar sir. From delegates, if there are any queries, any questions, any doubts, any difficulties, this is very good and very, I suppose it is very useful topic that using chemical fertilizers can be controlled and can be stopped for the betterment of mankind, for the welfare of humanity, for welfare of all living beings. In this connection, if you have any queries, you can ask directly to the resource person. You just unmute your mic and ask the question. I will wait for 2-3 minutes so that the questions will come, and Honorable Principal is on the way to join. Are there any questions? If any question from delegates, please ask your questions to Honorable Resource Person Pawar sir. The chairperson of this session, our principal, Honorable Dr. Nimburu sir, will conclude the session. Meanwhile, we are waiting for 2 minutes. If there are any questions from delegates, please ask directly. दीक्षित सर बाहेर क्लिअर आहे. दीक्षित सर फक्त उद्घाटन केलं त्यांनी उद्घाटन केलं 2 नाव हां आणि त्यांचा विषय हा आहे जैविक खतांचा वापर करायचा कमी करायचा तेव्हापासून Are there no questions? Should we go to conclusion? I request Honorable Principal Dr. Nimbore to conclude the session.
Thank you, Shinde sir. Good morning, all of you. Now today we organize one-day national webinar on emerging trends in agriculture biotechnology. Firstly, I will give my sincere thanks to Honorable President Kishore Nana Hambarde Secretary Atul Shetji Meher for give the permission to organize such a nice webinar. Then I give my sincere thanks to Honorable Professor Dr. P. Dixit, who inaugurated the session, and today's eminent person, Dr. P. Pawar sir. Now, in this session, it was the really nice session on emerging trends in agricultural biotechnology. I think this is the second time we got opportunity to listen to Honorable Dixit sir. In COVID pandemic also we listen Honorable Dixit sir. The resource person Honorable Pawar sir from Venkatesh Arts, Commerce and Science College, Deulgaon Raja, District Buldana, was very systematic. His discourse on plant growth promoting rhizobacteria for sustainable agriculture was very useful and informative. We must get a lesson from his speech and stop chemical fertilizers. Use of biofertilizers be promoted. Professor Khose from Department of Zoology and all organizing team took an initiative to organize this webinar. I thank all delegates and all of you. Thank you. Thank you.
Thank you, sir. I request Honorable our colleague Khose sir to propose vote of thanks. Professor Anand Khose.
Good morning to all. I am here to propose the vote of thanks. The first of all, I would like to express my sincere vote of thanks to our President of Asti Taluka Shikshan Prasarak Mandal, Honorable Kishorji Hambarde, to give permission to organize this one-day national webinar on emerging trends in agricultural biotechnology. I express my sincere gratitude, Honorable Dr. Dixit sir, his guidance in inaugural section. It was very nice to listen him after so many years. I also thanks to Honorable Resource Person Dr. P.B. Pawar, the Head of Department of Microbiology, Sri Venkatesh Art, Commerce, Science College, Deulgaon Raja. I would also thanks to Honorable Principal Dr. S.R. Nimbore sir, Advocate B.D. Hambarde Mahavidyalaya ST, and also Organizing Secretary Professor S.S. Moholkar sir, the Principal of College of Food Technology ST, for his support. I also like to thank our colleagues, my colleague Professor N.N. Nanote, IQAC Coordinator, and Dr. R.S. Sadbhai sir, IT Coordinator, for his technical support. I also thanks to Professor M.K. Kale sir and Professor Manoj Pawar sir from College of Food Technology for this technical support. And last but not least, I thanks to all the participants and delegate students for joining this webinar. With the permission of Principal Dr. S.R. Nimbore sir, I declare that the webinar is over now. I request all the participants to fulfill the feedback form. I immediately send the link of feedback form on a WhatsApp group. Thank you so much. Thank you. जेवण घेतलं